in working at Invivoscribe, and let's go to the next slide, please. Oh, wait, I have control. Is that when we work with pharma partners, we spend a lot of time helping them with their challenges. And so first, I'm just going to introduce our company a little bit for those of you who don't know who we are, and then I'm going to go into some of the challenges that we've run into that I think are easily avoided and can really save a lot of money and a lot of time when developing a targeted therapy that requires a companion diagnostic. So Invivoscribe, we are a diagnostic company. We focus on leukemia and lymphoma. We do crossover to solid tumors some. And we do our development and manufacturing out of our headquarters in San Diego. So another branch of what we do, as you can see there's international locations, is we have clinical labs that support both commercial testing as well as support doing the clinical trials that are needed for developing drugs. And so we have labs that are CLIA-CAP here in San Diego, and then in Germany, we have a lab that's DAX accredited, and in the Tokyo area, it's actually Kawasaki. We have a lab in Japan, and then we have a lab that supports clinical trials out of Shanghai, China as well. So, as noted, the first thing we do is we develop and manufacture diagnostics, and so we do have customers, roughly 700 customers worldwide, and this diagram does show that where the sites are that I had just mentioned, as well as the fact that we have distribution sites in both France and in Japan. So what are we going to talk about? What is the number one goal? So the agenda items are goals, regions of interest, the typical budgetary constraints that we run into in these sort of programs, and then issues that are caused by not considering the companion diagnostic early on, and then we'll just go into a synopsis. So as I go through issues, I'll also make recommendations. So some of it's long-winded, sorry, but I think it's really important stuff because the issues I'm going to be noting come up again and again, and it's not with small pharma or medium pharma, it's with large pharma too. So with that said, the number one goal when you're doing a targeted therapy is to get the targeted therapy approved because you want to treat patients. And the number two goal is to get the companion diagnostic approved because the targeted therapy is linked to the companion diagnostic since, as you already know, it's required to select patients for prescribing of the drug. And with all projects, budget is next. So you've got to stay within budget. And a lot of times the targeted therapies do not consider a realistic companion diagnostic costs, and that becomes a really big challenge for them later. And then also budget is impacted by region. So I chose a few example regions in the West and in the East. I obviously can't do a presentation that goes over the entire world, but just to show things that should be considered when doing a companion diagnostic that aren't always considered. And obviously, you could expand it to other regions as well. And then stay within project timeline. So some of the same things impact project timeline. So if you do good planning and you meet with the companion diagnostic company early, then of course, setting up timelines that are realistic is an easier thing to do. So we're going to talk about using the CDX in phase 2-3 enrollment, and we're going to talk about if a CDX is unavailable, what our recommendations would be for using clinical trial assays, and recommendations around setting up a proper environment so you'll have a successful bridging study should you use a clinical trial assay. And then we'll talk about regional time constraints because different regions have different time constraints. Most people know what the US is, I mean, in the submission times and the development times, but some of the other regions can be a mystery to companies because they really focus on the drug part and they don't focus on the diagnostic part. So I'm going to start with the West and regions of interest and things that I really think pharma companies need to consider and they don't consider when they're starting development in the US. So one is when you bring a companion diagnostic into the drug program, you have to introduce it to the FDA. And that's what the Q-Sub program's for. So the Q-Sub program, you get to both introduce the program and who your partner is, and you also get to show what your analytical strategies are, and what your clinical strategies are. So that's how you do it to CDRH. And then it should also be done to CDER. And with CDER, there's typically a type B meeting. And a lot of times the drug companies will hold this and not bring their diagnostic partner with them. And I think it's very useful to bring a representative from the diagnostic company with them, because CDER will bring a representative of CDRH with them. And so when you need someone to answer questions on some of the development challenges, it's useful to have an expert that's helping you with that meeting. So after you've done these introductions, the FDA is really, really good about confidentiality. They will not share across companies, and that's something I respect, and not something you have in all countries, which I will note as we go through this. But right of reference letters. So remember, you're going to have to write right of reference letters, and the earlier you do that, the better, because the earlier you do it, the quicker CDER and CDRH will talk about the two different companies' programs together. And that kind of helps alleviate things when you get to submission time, so you don't have less deficiencies. And so let's talk about US and timeline. So classical PMAs, 180 days. That's assuming you don't have any severe deficiencies that are going to cause a clock stop. But the US is also flexible, and this is something that I really like about dealing with the US FDA, in that typically with a companion diagnostic program, it's common to do what's called a modular PMA. And that is something that should be considered early on, because you have to do submissions specific to requesting a modular PMA and presenting that strategy and what the modular shell will look like to the agency. And so again, it's something to remember to include in your timeline, and that'll typically switch from 180 days approval to 90 days approval. So you really do bring in the timeline. So, and if you have something like breakthrough therapy, it does reduce the risk of the device holding up the drug. So this last one isn't really an issue of any sort. I just wanted to note, because a draft guidance actually just recently came out by CDRH with regard to co-developing drugs and CDXs. It's really good. So I'm just going to note, read the draft guidance because it helps, and I'm going to go from there to the Europe. So in Europe, the in vitro, so first off, targeted therapies, at least in my world, because I work in oncology, they a lot of times require companion diagnostics. And in Europe, it was covered under the in vitro diagnostics directive. And that directive meant self-declare for most oncology products, which was basically the Wild West and very easy. And they just changed it completely. So now they passed the in vitro diagnostic regulations that replaced the in vitro diagnostic directive. And I'm sorry if that's a bit of a tongue twister. And the rules are far more restringent. You have a lot more submission requirements, a lot more validation requirements, and obviously a lot more impact timeline and budget. So having to meet the in vitro diagnostic regulations is very important, and now manufacturers to do CE marking don't just need ISO 1345, but the developer manufacturer also needs to be accredited to the in vitro diagnostic regulations. The IVDR reviews right now are typically slow. That they're supposed to take 6 months, but they're typically taking closer to 12. And that's because not a lot of notified bodies are even set up to review these IVD submissions, especially companion diagnostic submissions. And so there's a lot of training going on in the EU right now to get notified bodies up to speed so that they can support the IVDR. And then I'll have to admit, COVID hit too. So they've got a brand new system. All the self-declares have to submit. So you've got all these international IVD manufacturers submitting at once. And then all these COVID assays also showed up. And though this isn't a region I'm going to focus on, I am going to note one of the sad changes to the EU is that the UK is no longer included in CE marking. So now you have to get a UKCA mark, which you have to submit directly through the UK to get. It's similar to CE marking, but you do have some other hoops you have to jump through. So, and what I didn't, I skipped one. So in the US, you submit a PMA. So all companion diagnostics are considered high-risk IVDs, and so that's a class 3. And you'll see this as a theme. So in Europe now, under this new IVDR, you have a class C CDX submission, and it's the same thing as class 3 in the US. It's a high-risk submission, and very similar to the US, they're now doing joint review between the drug and the device side. So you pay for a joint review between the notified body and the EMA, which is new, because obviously when it was self-declare, they weren't doing that. So Japan. This is a market that typically people see as secondary, and I hate saying that if there's somebody here from Japan, but they typically go for the US, EU together, and then they jump and they go, okay, I gotta add Asia on. And Japan is one of the first markets to go to because it's very friendly to the West, and the submission process isn't, in my opinion, that difficult. But one of the things to consider, and one of the things we note, is that you have to have Japanese subjects either in your phase 3 development program of the drug, or you're going to be doing a confirmatory study. So in Japan, again, it's very similar to the US and Europe. It's a class 3 product, and you do a PMA-ish submission. I don't know how many people are pharma here, but PMA is pre-market approval, and that's a class 3 submission to the US FDA to get approval. Shonin is the same thing to the PMDA, which is the equivalent to the FDA in Japan. But remembering, when you're figuring out budget and timeline, there is completely different formatting and translation and labeling requirements in Japan than there are in the US. Also, just like the US, Japan is very good about negotiating. One of the things I like about both the US and Japan is the science is what drives everything. And so you can negotiate with the FDA and the PMDA to come up with what are the best studies to basically validate an assay. I'm going to give you a reason for not doing that too, which is kind of weird, but I will. And so in the US, I mentioned Q-subs. In Japan, you do in-person consultation meetings. Everything's face-to-face in Japan, which is fun in one way, but another way it's kind of inconvenient because if you don't have a team in Japan, you're flying back and forth to Japan. So why would I say I like science-driven PMDA and FDA negotiations, but say that it's not always the perfect path forward? That's because you could negotiate a less onerous pathway with those two organizations and get approvals. But Europe and China's my next slide, they're more checkboxers. So if you didn't follow CLSI or an ISO standard very specifically, you won't meet the checkboxes. So you've come up with these great solutions and you negotiated them with agencies, but they don't meet the checkbox in other regions, and then you end up having to do more studies because of deficiencies in your submission. So that's just something to note. Japan also has some specific requirements of their own, like their stability requirements are a little bit stricter than others. You have to do your shipping study and your open stability, and your full-length stability is kind of a combined thing in Japan, which you do not have to do with the US FDA. So it's just an example of kind of regional specific things you have to pay attention to, and you want to have a partner who knows those regional specific things because that helps. And in Japan, you do have to have a manufacturing authorization holder and a manufacturer who's registered their quality management system with the PMDA. So obviously, Invivoscribe has done these things, but so have other companies. So another thing to note in Japan is business model. So you cannot, even if you've got the drug and the device approved, you cannot commercialize until you have reimbursement for both the drug and the device. And also, they want to make sure you have a proper business model. So be it a single-site assay or distributable assay. Each region does it differently. Like in the US, there's a lot of central labs. And in Japan, there's also a lot of central labs. But in Europe, if you tried to use a single central lab in one country and have all the other countries shipped to it, it's not going to happen. They want basically an assay in each country typically. So I typically suggest going to a distributable model because it opens more regions for you and makes it so you don't have to do later development to a distributable model. But As noted, you have to deal with the MHLW Economics Division after getting approval to get your reimbursement, and it's another thing that takes time and is not always considered. So China. It's actually kind of funny. The first two words on my slide are very difficult, and that's actually very true about China. So China is a huge market and a growing market, and one that I suggest everybody goes to, but at the same time, I think they're actually easier for drug than they are for device, and I would say that's very atypical. And that's because the CDE, which is Center for Drug Evaluation, which is part of the NMPA, which is their FDA, they communicate very similar to the FDA does with the pharma partners, whereas the CMDE doesn't have a very open pathway to negotiate with device companies. So really what it comes down to is you have to follow letter of the law. And their regulations are revamped annually, and the development programs take years. So note, when you go to China, before you get approval and you've come up with your development program, the rules will change. And so you will have to be a little bit dynamic and adjust to the rules in China, which is tough. So just like Japan, China wants you to test in their patients when you're doing the drug development. Now, where it's different is they require you to test the IVD in their patients. So pretty much all regions in the world, I think we have a lot of IVD people in the room, will require an IVD clinical accuracy study where you're testing with the investigational device and a reference method, and you're doing positive percent agreement, negative percent agreement. Well, you typically do that once, or you do it for all your different specimen types. And then you can submit that all over the world, except for China. So in China, they will have you run a clinical validation of the IVD separate from the drug. And so that is something to consider, because that typically is the rate determining step in China, because that program usually takes a couple of years. And you have to run it in AAA hospitals. So there's a lot of things you have to think about, like contracting with the hospitals, getting all the various ethics approvals. We're in a next generation sequencing conference here, so remember it's human genetics, so you have to get the human genetics research approvals as well, which are actually kind of challenging, and there's multiple different types of them. And then you have to identify a Chinese-specific reference method. So a lot of times you might develop your own reference method or compare to an approved product or an LDT out there. Well, in China, you definitely have to choose a method that, if you're novel, is an LDT run by one of the AAA hospitals. And there's a lot of other things, but I'm not going to get into that. And just to note, again, as a theme, Class 3 CDX, so high risk. And they, very much like Japan, have their own format, translation requirements, and labeling requirements. And typically, they have a 12-month review cycle for a companion diagnostic. So also, you have to have a authorized registration agent within country to manage your submission. As noted in an earlier slide, we have a site in Shanghai who represents us for doing these sorts of submissions. So, all right, so that gives you an idea of just kind of the, I guess, east-west regional things that I note that pharma companies don't always take into consideration. But they always take budgetary things into consideration. So let's jump into budget. So a lot of times, I think with a drug program, you know, it takes 10 years or more to develop a drug, and you don't want to release budget until you get to phase 3. And so you have these issues where by the time they contact the CDX company, they haven't released budget yet, and they waited so long, there's no time to develop the companion diagnostic. So what we have is CDX costs too much, and the drug may not validate successfully. So and that's a lot of times because they wait until phase 3 to contact a companion diagnostic company. And there are ways to structure deals so that you don't cost too much for the drug developer. And then there's the other bit, which is clinical trial assays. When I say clinical trial assay, that means it's a companion diagnostic that's not the companion diagnostic that's only used in like the phase 3 trial to select patients. I guess it could be used in phase 1 too. But these are typically laboratory-developed tests. And yeah, the drug companies like using them because CDER never says no, and they're typically quick and easy to set up. And I'll get into reasons why I would suggest either not using a CTA or just being very careful when using them and limiting how many you use and checking the performance of them before you use them. This one is huge. So controlling specimen, sample processing, and storage is difficult and expensive. So I'll get into that in a later slide. But I've talked with so many either pharma partners that we're already partnering with or potential pharma partners where this has been a challenge because it wasn't done right early in the clinical development of the drug, and then it impacted them in a negative manner. And so, again, with controlling your trials right, there's a way to manage through this. So vetting CTAs, so clinical trial assays, difficult and expensive, but I already noted it should be done, and I'll explain more about that in the slides that follow. And then if you're going to be doing a targeted therapy and you're going to be selecting patients, then you probably have a biomarker in mind. And if you have that biomarker in mind, you might not have it fully vetted yet, like what specific mutation variants matter or what your clinical cutoff is. Again, when you do an IND, since they're safety-based, the agency might let you move forward with your trial, but you might not have a validatable CDX because you didn't do this part, or a way to explain that you did have drug efficacy or selected right because you didn't do this part ahead of time. And I'll get into that more as we move forward. So, common issues. Well, I think I already noted this one. So pharma contacts the companion diagnostic company too late. And so you got to remember these agreements are usually between two big companies with legal departments, and they take months and months to negotiate. So not only is contacting a companion diagnostic partner late not recommended because you won't leave time for the development program, but even if you contacted them in a time where you thought you did leave time for the development program, if your contract gets caught up in legal, you might be burning through your timeline and then pushing out the approval time for the drug, which is awful because drugs, well, they can make millions per month. So I guess that's a good financial, but at the same time, you need them to treat these patients because we're talking about oncology, at least I am in these examples. And with oncology, these are very sick patients and we want to get these new therapies to them. So I threw timeline in here because it's directly related to budget, but at 2 to 3 years from inception to submission for a companion diagnostic, like a next-gen sequencing assay, I would say is a fair estimate. And so if you think about, oh, I'm a pharma company, I'm going to start enrolling people in 6 months into my phase 3 trial, and then you go and approach a diagnostic company, you're putting yourself in the case where you're going to have to bridge because you're going to have to run a CTA and then develop the CDX in parallel. And avoiding bridging removes barriers with the regulatory authorities. So as noted, that'll impact subject selection readiness and may require bridging strategies. So also, when you meet too late with your companion diagnostic partner, you'll want to truncate things. You'll want to do quicker studies or simpler studies, not as many replicates and that sort of thing. And so there's just too much incentive to cut corners when you do that. And when you cut those corners, it sounds good in a core team, and it might work, like I said, for certain regions, but not other regions internationally. That's one thing. Remember, you want to study each of your studies to work across all countries, not just one. And so it's something to be avoided. And if you had an appropriate timeline, because you met earlier, then you would have a lot less incentive to truncate things. And then, as I noted before, a clinical trial assay is available to fill in a lot of times in place of a companion diagnostic, but it does increase the risk of discordance. And if you have discordance, because that forces a bridging study, well, a bridging study where you don't get the proper agreement results in non-approval of the CDX, and since the drug needs the CDX to get approved, it's just not a very good outcome. So obvious recommendation is approach the companion diagnostic partner as early as possible to avoid drug approval delays. So this is one, it's funny. So I got called by a friend at Thermo who said, oh, I hear you're talking, and you're talking about common issues. And I was like, yeah. And they're like, are you bringing up negative samples? And I'm like, oh yeah, it's already on the slide. So everybody knows who works in the diagnostic realm and has helped with companion diagnostics that one of the biggest challenges is getting clinical samples. And that's because the handling of clinical samples, the storage of clinical samples is typically not done right. And then it ends up costing a lot of time and money. So we'll get into that. You need your phase 1, phase 2 subject samples stored appropriately to support setting the cutoff and doing analytical validations of the companion diagnostic. So how do you avoid not stored properly? Well, some of this can be mitigated, in my opinion, by thinking about the companion diagnostic during phase 1, phase 2 development. And so write your protocols so they're stringent about what sample types, and they're stringent about how you process and store them. So I will note, you have to be specifically mindful when you're dealing with things like RNA or expression assay samples, because both of those will degrade or change very quickly. And so before you even go into phase 1, you should probably spend a decent amount of time developing a process to ensure that your RNA or expression assay samples have a storage method that will actually store them long enough so they'll be stable and will give a similar answer as your CTA did when you go to run them over a CDX later. So another thing that you run into is the demographic data. So I think phase 1, phase 2, why keep demographic data that it relates to a companion diagnostic? And it's because, well, you're gonna need it because you're gonna wanna know that they're the right samples to be developing the companion diagnostic with. And you don't just need it for the positives. That's another thing. In a drug study, the negatives don't enroll. And so they don't enroll them and then they don't get demographics on them. Well, you do need a few basic demographics because you're gonna need it for the diagnostic, because the diagnostic cannot be validated without negatives. The positives and the negatives are equally important to a diagnostic. And then not the same specimen type is planned for the companion diagnostic. Well, there can be a couple of things that you run into here. Either a completely different specimen type was used in phase 1 and phase 2 than in phase 3, or you could run into something where, oh, we told them to just freeze the cells down or freeze the blood down. Well, I'll tell you right now, when you're dealing with regulatory authorities, if you tell them that you got blood or bone marrow and you extracted the DNA and you stored it at -20, they'll let you call that blood and bone marrow later when you go to test them with a diagnostic development program. But if you froze the cells down, they're going to say, that's a different specimen type, and we expect that'll have completely different performance than the original blood and bone marrow, which means you now have to validate 2 different specimen types. You have to validate a frozen specimen and a fresh specimen. So these are the sort of things, again, you need to consider. I even threw in anticoagulants in here because it can impact whether you have to do equivalency studies or add stuff to your interfering substances. But very much an important thing to consider that, again, drug companies don't consider. So what they'll run into is doctor says they always store it this way, or it costs too much to extract all the DNA and store it. And so they'll get kind of bullied by their clinical sites while they're trying to save money in the phase 1, phase 2s, and they won't write stringent clinical protocols and have monitors that are forcing people to all do it in a consistent way. And I'm going to say, make sure it's done in a consistent way, otherwise you don't have consistent samples for doing the companion diagnostic. And the way around that, we'll get into, well, I'll just say it, you end up having to do a collection. And so you have a whole nother clinical protocol where you're collecting positives and negatives, and nobody wants to have to do that. So, not processed appropriately. So this could be cell pellets, cell isolations, extractions, dilutions, whatever. So again, same thing as I was saying before. You're just going to want to control your clinical programs so that the samples are all handled exactly the same, so that you don't have a whole bunch of specimens to validate come companion diagnostic time. So, and I noted this, it may result in a protocol specific to collect positives and negatives for the assay development. So it's even worse when you do this in phase 3. So as I said, don't show up too late. Well, a lot of pharma companies show up and they've already done phase 3 with a clinical trial assay. So if you've done phase 3 with a clinical trial assay and you didn't store your negatives, or you had a bunch of specimen types, you run into a situation where, okay, I didn't store everything right. Now you have to do a specific collection protocol, and it has to be tested with the clinical trial assay, the companion diagnostic, and the reference method. So that's a nightmare. And again, here's a recommendation. Characterize and save as many samples as possible for potential future use with the CDX. So next. So use of multiple CTAs, so the clinical trial assays. Again, I'll note that this gets back into meeting with the diagnostic partner too late. But if you have to use a clinical trial assay, avoid using multiple assays, because they're probably going to identify different variants at different sensitivities, which means your companion diagnostic is going to have trouble agreeing with it. So the mitigation for this is to use as few CTAs as possible and to do vetting. So what I typically would suggest is, when choosing a CTA, contrive proficiency panels and make them pass those panels before you use those sites. So include, the variants of interest, and include variants that aren't of interest to make sure they're not calling those positive too. And include various dilutions around your clinical cutoff. So again, I would recommend that you either develop the companion diagnostic for the selection, or that you try to focus on using just a singular clinical trial assay, so you only have one assay to build agreement to when you develop the companion diagnostic. I know that's not always possible, but that would be the recommendation. So assay configuration, I already noted this one. So this would be like you did single site, but then you're going to go to another region like Japan, which is going to require you a distributable assay. Or you chose a technology that's really R&D, so it's really hard to turn into distributable later. So this is something to consider. Sometimes it's worth spending a little more money up front and just developing the assay correctly so it'll work in all regions across the world. So take time to think about your commercial needs so that you develop the assay once and you don't have to keep revalidating it or making changes to it. And then time to obtain regional reimbursement approvals. I already noted that as well. So the pharma companies don't always think about the device reimbursement, and in various regions, reimbursement is very much required for the device before the drug can be sold. So, all right, I don't know how I'm doing on time here, but hopefully well. Huh? Oh, shoot. So quick synopsis then. So these are just examples of common oversights that I've noted while working with pharma partners. And careful planning of phase 1 through phase 3 clinical development of the drug, I believe it helps avoid much of this. And also, I believe doing contracts where if the diagnostic and diagnostic and drug company, where you have milestones that build over time, allow you to have early engagement, but not spend all the drug company's money right away. So it can ramp up as the drug is showing more success, then you open up more budget and you do more and more validation of the device. And that's really my recommendation, is meet early and set up contracts like that, and meet early enough that you can come up with contracts like that. So always consider your regional requirements and costs in your timelines, because they are very different across regions, as noted in the slides that I showed already. And then it's really the drug company's responsibility to do this, but it really helps if they work with the device company to do it, but validate the biomarkers in the clinical cutoff and be very restrictive on the sample types that you use in your studies. And last but not least, don't forget about the commercial strategies and reimbursement, because they take time to set up and they might impact how you configure your assay. And I apologize for going a little over, but do you have questions for me? Oh,
we're breaking for lunch now, and we'll be getting started back here at 1:30.
All right, well, thank you, everybody, and have a nice lunch.